Okay, today we have Xesca that just started her PhD not long ago, and with Jonas. So lots of people from Jonas group. So thank you for presenting this afternoon.
Okay, so yeah.
Sí, no, es que esa aquí.
Ahora. ¿Sí? Vale. Good afternoon. As Tim already said, I'm a PhD student from Fiona's group. So today I'm going to give you an overview about what I'm studying, but with us focusing on one of the experiments that we already have been developing. So to start, what is my project about? Basically, this project is about to evaluate the influence of microbiome in seagrass health and their resistance to stress factors. And we can say that we have three main points, which are functionality, resilience, and the future application that we can have in restoration. And these 3 points are developed in 3 specific objectives. The first one is characterize the differences between microbiome from seagrass communities with different conservation states, just to evaluate their role in limiting the natural recovery. Then we have the second one, which is to evaluate the influence that these microbes have in seed germination and seedling development. And the last one is to explore the use of this microbes community in assisted evolution approaches. But as I already said, today I'm going to talk about the second one, which is the first one from where we have some data now. So this one is focused on a Mediterranean seagrass community from Balearic Sea. Here we have different species like Phociona oceanica, Tostera nolti, or Timothea nodosa. And especially we focus our sampling area in a close bay from the north of the island, where we mainly find Timothea nodosa midos, which is the species in which we use for our first experiment. So as you can see, everything is about seagrasses. I won't go so deep in the context. I'm sure that most of you already know how important and threatened these ecosystems are. I just wanted to leave here, of course, a small scheme about the different ecological benefits that they provide. And especially this map that seems very visual to me to represent how is the decline of different species around Europe for the last years. So given their importance and their regression, seagrass conservation is a global priority. And this fact had led, especially in the last years, an increased interest in restoration. And with this context, the need for projects that promote their recovery is clear. But until now, most of these restoration project activities had focused on the structure and extension of the ecosystem. And if we have into account that we live in a context with the continuous increase of different stress factors, for example, climate change, what we want with this project is to study the effect of the factor that can promote the resilience rather than just replicate the structure. And we decide to focus on the microbiome as one of these determining factors in understanding the resilience of these ecosystems. And why microbiome? Because I know there are different factors that are important for seagrass health and their resilience, but we want to study the microbiome because although the seagrass microbiome is still in its early stages, there are some studies that prove that they have a diverse microbiome that can contribute in some aspects of seagrass development and growth. So with that, and in addition, their effect in health for terrestrial plants have been already proved. So with that, and also taking into account that the seagrass for life stages can be critical for natural recovery, we start with this hypothesis that is that the presence of some microbes can affect germination and development in Timothea nodosa. And we want to answer some questions about which is the effect of this microbiome in seagrass germination and development, but also how these communities change for different treatments and along developmental stages, seed developmental stages. So for that, we carry on an experiment that consists in applying sanitization to seeds. We start with the field sampling where we collect some of the seeds that we need for the experiment, and then back to the lab, we select the viable seeds and distribute them in aquaria waiting for the setup. This experiment setup have 3 points, 3 steps. The first one, we sterilize 50% of the seeds with a combination of 20 sonication and bleach. And then we subject them, well, the seeds from both treatments, the sanitized and not, to a hyposalinity shock, because we use the hyposalinity shock method because some studies prove that this breaks dormancy, what means that they enhance germination and that reduce our experimental times, just for that. And after a week, we start recording the germination subseed for both treatments. Then we go with the second step, where we take the germinated seeds and plant them in test tubes, as you can see in the picture, to follow how many seeds grow to a green elongated cotyledon. And also during all this time, we follow the growth of the seeds and take sample for different stages. And then we have the last step, which is just the comparing of microbial communities by sequencing, the seediness in that case. And with all that, we obtain our first results. Well, first of all, here you have what I mean when I'm talking about germinated seeds, which is not when the seed is just open, it's when we already consider the cotyledon. And well, here you can see how sanitized seeds have low germination rates compared to the unsanitized seeds. Then after that, we follow the growth, the germinated from elongated green cotyledon, as you can see here. And more or less both treatments start developing at the same time, but at the end, there are more unsanitized seeds that grow than the sanitized. And I have to point here that don't take so attention to the time, because we see some kind of delay because of the method, but we can talk about this method later. But the important thing is at the end, there are a difference between treatments. And the end is the microbiome. First of all, about the richness, we see that there are significant difference between sample type and treatment. And of course, there are more richness in field samples, and also the richness is higher for unsanitized seeds compared to the sanitized seeds with the same developmental stage. So now we know that there are differences about richness, but we want to see how similar or different these microbes communities are. So we are going to check this figure in different parts. First of all, we can see, of course, that the agarose for both treatments change from the time 0, where the seed is just planted, and when the seeds are growing, of course, the microbes are colonizing the agarose. Then we can see that the unsanitized seeds for all the developmental stage are more similar to the field. And also, they are more similar, the unsanitized seeds from different developmental stages have more similar communities. But if we check the sanitized ones, of course, they are different from the field samples. But also, when the seeds grow, they change the community in some way. And also, this is changing in a different way to the unsanitized ones. So there are some differences between microbial communities. And this is interesting thing, and that is why I want to finish with this figure, because we know that the richness is different. There are different germination rates for both treatments, and also the microbial community is different. And now we want to know who is driving these differences. I can't go so deep with this figure because I didn't have the time to check deeply who is driver or the mechanism that this taxa is using, but we can see some difference about the abundance. And also here we have some nitrogen fixator and symbionts that can promote growth hormones or something like this. So I hope that we will do a little more study about that. But at the end, what we were asking is that if the presence of these microbes have an effect on germination, and we can prove that with these results. And also that the microbial community are different between treatments. And well, what's happening now? Of course, we are analyzing field samples to characterize how the communities are different in the field, but especially we are testing different methods to repeat this experiment with more accurate data in that case. And this is.
Well, if I understand well, you have done experiment where you have seeds that have been treated, you try to sterilize them, and those that have not been treated, and you see a difference in the germination rate, right? And you attribute this difference to the presence or absence of the microbial community around the seeds. So it could also be that you're just disrupting something in the seeds during the process of what do you call it? Let's say the preparation, the sanitization.
Yeah.
Okay.
That's the right.
So I'm sure that you have discussed this. Have you tried, for example, take sanitized seeds and sort of rub them with the microbial community, where if you just touch and touch again, touch the untreated and you touch the treated, you transfer the community. So have you tried to see whether there is a difference in the growth between these three?
Yeah, that's what we are trying right now. We did some tests where we have sterilized seeds that we put together with unsterilized seeds and also sterilize it with sterilized, just to see how they are growing. And I don't have the data because this is something that we are doing now. That's why I say that I want to improve this method, because I wasn't so happy about the bleeds and these things. And they are growing good. I mean, I don't have the number of the data, but they are, I mean, they are not dead at that point. So it looks like yes, but I can't say because I don't have the data.
Looks like yes, meaning?
I mean, I have seedlings. The sterilized seeds are growing, so they are alive. But how?
You don't know whether.
I don't know about the microbial community. I mean, I know that I sterilize them, I put together again with another seed, non-sterilize it, and they are growing. No. Is that what you are asking? No, okay.
I was asking whether let's say the sterilized was growing differently than the sterilized non-sterilized.
No, they are all of them are growing. I mean, they are growing.
But then if you put together the sterilized and non-sterilized, the sterilized one would be I need to sleep more. Will be colonized by the microbial community of the unsterilized. And so if now that grows the same as the sterilized, then you can't attribute the differences in growth to the presence of microbial community.
Yeah. I mean, yeah, I know what you mean. I can't ask that because this is something that I am doing, so I don't want to say yes or not about that, because we are checking that, and also we are trying, because for me the problem is especially the agarose in the growth. So we want to see also about the nutrients that the plant have and everything, so and how big they are, because they are germinating, but maybe not that big or not that strong. So this is something that we want to analyze, but I did not yet.
Okay. It's about the basis of the study, or the end of the study. And again, I'm not an expert, so I may not understand some of the things, but my side is it would be obvious that a community with bacteria that are actually generating certain metabolites and certain substances available to the plant would be all that's what I would expect. Anyway, one of the things is that I don't see or about your forward work is how would you transport this to the actually to the nature, to the open field,
Yeah.
because you are growing this and under very specific conditions. But I would assume that the microbial community in the field is not solely based on interactions with the plant, but particularly is the effect of the type of soil, the organic matter, etc. So how do you jump from your lab experiments to the field?
Yeah. First of all, for these experiments, I had more treatments, but I mean, the agarose was the problem. And we use sediment microbes from different environments just to see which environment it's better to restoration, talking about the impact of microbes from this area. But I lose these treatments for a lot of reasons. And this is one of the things, just to do the same experiment, but using soil microbes, just to see, okay, this kind of soil from this area with this, also checking about the nutrients of the soil, because I have an important effect on microbes. So we want to apply this treatment to see which kind of soil can be better for restoration. And then we are thinking about, because I just started, so I'm thinking about things, but I need more time to do them. In Mar Menor, they have some differences between germination from different areas. So maybe take seeds from bad area to a good area and see if they are growing there and the microbes are changing to the good area community, if this is the effect, you know what I mean?
Right.
Yeah,
Yes,
this is something that we are like trying to figure out to transport to the nature.
Although I anticipate that it's quite complex because it's not just the bacteria community, which is obviously important, but also the sediment grain size and probably the oxy conditions that make it available. For example, iron probably is very important, and that depends on the oxy conditions of the sediment. So the biochemistry there is quite complex.
Yeah, and this is something that we are trying to put all together. But this was just like a first trial to see how the seeds are reacting.
It just continues with the question of Watson, that I think that maybe the point is that to know the amount of biodiversity that has the microbiome instead of go deeper into that with that microbiome works and this other. Your idea, I don't know, I'm asking you just.
Yeah, I mean, it's quite difficult to say this taxa is the one that is working. So we are working with all the community together. So which kind of community? I mean, I cannot say this bacteria exactly is the one that is driving this, because this bacteria in another environment maybe is not working.
But maybe you shouldn't look for bacteria, maybe you should look for the metabolites instead of bacteria. Because bacteria is the richness of the community is very, very difficult to compare with other one side to other. So maybe different bacteria in different places may be actually doing or have the same or producing the same stuff. So maybe it's easier to look for metabolites or for certain micronutrients, etc. That maybe you may know that they are beneficial for the growth of possible.
Yeah, this is something that we want to check especially in the sediments and in the water that the plant is using, because maybe I have a bacteria that is giving some kind of nutrient, and I can check that. So in the next experiment that we want to repeat, I don't know how yet, but yeah, we want to start like taking all the factors into account.
Yeah, I have two quick questions. One is following Marco's
comment, have you thought about using antibiotics instead of bleach?
I'm using antibiotics now. I mean, because bleach don't look like visually kill the seed, but we can't prove that 100% for all the seeds. So we are using now antibiotics.
And then the other question is, I think it was your second slide, you showed the objectives of your project. The third one, was about the assisted evolution or something like
Well,
that.
yeah, it's to use what we learned from this experiment that, oh, this environment or this soil have this community that enhance germination and growth. So let's try to use this to make the restoration, you know, that's.
Yeah, one question about, well, maybe it's a technical question from my lack of knowledge about this thing. Because this microbiome, it's associated to the seed, or is the soil microbiome? I mean, it's the same, or there are two different microbiomes?
They are different.
Okay.
Yeah.
So it's associated to seeds, not only.
In this experiment, yes, but that's why it's important also to add.
Wait, no.
That's one. Thank you for the talk. I'm probably gonna ask something really stupid because I have a terrestrial brain. So I was thinking about the seeds,
It's a revelation.
and whether there has been any report of seed dispersal by animals in the sea. So whether this could be something that has been studied somewhere, because I've never seen seeds in the sea and how, I assume they dispersed by water. But in land, it's something they
Yeah.
have an advantage if they are dispersed by.
I mean, Timothea's seeds are buried, en plan enterradas.
Yeah.
In the sediment because they appear to close to the rhizome with the sediment. So we find them under the
So
sediment.
they are quite deep.
Mhm.
So there's no plant that has like the seeds.
Yeah.
In the, there's no reports on dispersal like fish.
No, they had, what,
No, I don't know.
they had some bites from sulfites, but no.
Timothea?
No,
Okay, I was saying about Timothea.
no, because she asked if there is some of the seagrasses that have
Okay.
no flower.
Yeah. That could be quite.
Well, we just come in one campaign, we persecute sulfites with the bag when they were full. There we were there, and they have some algae, but more,
There's something.
yeah, and leaf and rest of leaves, but no seeds, and sand.
Posidonia.
Thank you.
Yeah, all good. Thank you, Xesca.